Okay, so let's move on to our second class of macromolecule, and that's going to be lipids. So, unlike carbohydrates, lipids are not water soluble, and this is a characteristic that really is going to shape their biological role. So, from energy storage to membrane structure to hormone production, lipids are really, really diverse, and they actually do not have a true monomer because you'll see the different structural variations. They're not an individual monomer that we link together. So lipids are a broad class of hydrophobic molecules, again, meaning they do not dissolve in water. This is due to their structural differences. So they have long nonpolar hydrocarbon chains and a relative lack of hydrophilic functional groups, meaning they don't have functional groups that are attracting water. So while they seem like simple fats, lipids actually include a huge range of molecules with different functions. So we have fats and oils for long-term energy storage. We also have phospholipids, which are going to make up our cell membranes. And then we have steroids, which are for signaling and regulation. So again, they're all very structurally different, so there is no true monomer when we're talking about this class of macromolecules. So the most familiar lipids are going to be fats and oils, which are typically built from two subunits. So we have a glycerol, which is a 3-carbon molecule with 3 hydroxyl groups attached to it. So this is our glycerol here. And then we have fatty acids, and these are just long chains of carbon and hydrogen with a carboxyl group at one end. So this is our fatty acids. If we're talking about a triglyceride, it has 3 fatty acid chains and 1 glycerol at the end. This is the carboxyl group, or the COOH that they were talking about previously. So when we have these 3 fatty acid chains that bind to the 1 glycerol, it's going to give us a triglyceride. And just like we talked about building all of our other macromolecules, this reaction to form it is going to also occur via dehydration reaction, where we're going to have to remove 3 water molecules, 1 for each of the fatty acid chains, in order to link it to the glycerol itself. So triglycerides are incredibly efficient at storing energy, and they pack more than twice the energy of carbohydrates, which is why our body stores them for long-term use. Oils also play a role, so things like waterproofing feathers, our hair, or even our skin if we're talking about animals. So fatty acids can either be saturated or unsaturated, and this difference is really going to affect their structure and function. So if we're talking about saturated fatty acids, they do not have any double bonds between carbon atoms, and this results in straight chains that pack tightly together, making saturated fats solids at room temperature, kind of like butter. Unsaturated fatty acids have 1 or more double bonds in their carbon chains, and this causes little kinks or bends in them that prevent tight packing. So unsaturated fatty acids, unsaturated fats are typically liquids at room temperature, so things like olive oil. We also have a special type of unsaturated fat, and that's going to be a trans fat, where the hydrogen atoms around the double bond are actually in opposite sides, and this is going to make the molecule more linear. So trans fats are actually artificially produced, and they are the ones that are linked and associated with health issues. So phospholipids, these are really important lipids that play a role in forming biological membranes. So if we're talking about a phospholipid, it's going to have a polar phosphate head, and that means that it's going to be hydrophilic or water-loving. It's also going to have 2 nonpolar fatty acid tails. So here's one, here's the second one, and these are hydrophobic, meaning water repelling. But if you've noticed, it actually has a dual nature. So that makes phospholipids amphipathic, meaning that it has both a hydrophilic and a hydrophobic region. So in water, they spontaneously form bilayers, which is basically the foundation of cell membranes, and it's going to help keep the inside of the cell distinct from its outside environment. So this is why lipids do not have a true monomer, because shown to you in the images on the right are another type of lipid, which are steroids, and they look very structurally different than the phospholipids and the triglycerides that we just looked at previously. But they still share some of the common characteristics of lipids, for example, being hydrophobic and nonpolar in nature. So steroids are going to be composed of 4 fused carbon rings, and even though they do not contain fatty acid chains, they're still classified as lipids because they are not soluble in water. The most well-known steroid is going to be cholesterol, and it's going to serve as a precursor for steroid hormones like testosterone and estrogen. It's also a precursor for vitamin D and bile salts, which are going to help digest fats. So steroids are going to differ from each other based on the functional groups attached to the ring structure. So small changes can actually lead to major differences in biological activity.